All right, so good day, everyone. Today we're going to have the discussion of the fifth experiment in the biochemistry of the gene laboratory, which is entitled the antigen detection using enzyme-linked immunosorbent assay, ELISA. Okay? So this will be the outline of this discussion. So I'll be discussing first the introduction as well as the background of ELISA, and then that will be followed by the objectives of the experiment, followed by the methodology rundown. Okay, and then of course certain considerations lang in the methodology as well as the references. So what is ELISA? Okay, technically, before we go into the detail of what the method is, so we will have first a very quick view of our immunity. Okay, so immunology is the science that is concerned with immunity, the study of our immunity, as well as the biochemical basis of our immune system. So technically, how our immune system fights certain disease, okay, in terms of biochemical aspects. So it deals with the physiological functioning of the immune system and stating both the health as well as the disease, as well as the malfunctioning of the immune system as a whole. Okay, so this covers certain concepts like allergies, immune deficiencies, as well as autoimmune disorders. Okay, so in the study of immunology, so the molecular biology principles are also incorporated. So here are the targets of the immune system. Okay, so first and foremost, of course, the invading pathogens in our body. So examples of these are viruses, bacteria, parasites, fungi, and protozoans that maybe intentionally or unintentionally has entered our body. Okay, and these particular pathogens, okay, can cause a certain disease. Okay. Next are the worn out cells. So these are cells, they are your own cells. And if the body is unable to remove this worn out cell, they can result to certain dysfunctions, such as formation or triggers of cancers and tumors. So next are the mutant cells. So mutant cells, these are your cells, okay, that has develop, okay, a certain change specifically in their genes leading to their abnormality. Okay, so this includes abnormal cells in the body, and they may be growing rapidly, uncontrollably, etc., etc. Okay, so this may cause a certain problem in a particular tissue or organ if not removed. Okay, and then of course we have the cellular debris. So cellular debris, these are the remnants or remains of broken down cells um, that has been eliminated. Okay, so these cellular debris, they can be detected by certain cells, and um, may cause also certain problems in the body. Okay, so the immune system is actually composed of two different parts. We have the innate immune system and adaptive immune system. Okay, so in innate immune system, so this is the faster portion of our body. So its response is non-specific. So regardless if there's a pathogen or if there's an infected cell or whatsoever that causes certain problems in the body, okay, so the innate immune system will react. So the exposure leads to immediate maximal response. Okay, so again, immediate response siya, okay, to the certain pathogens. Okay, so this can be subdivided into cell-mediated um, response. Okay, those that are done by your white blood cells. Okay, um, as well as humoral components, such as those of the complement proteins. So the innate immune system also bear no immunological function. Okay, so therefore they cannot remember if a particular pathogen has already entered the body. As long as the pathogen is detected, ayun, they will remove it from the body. Okay, and this innate immune system is also found in all forms of life. Okay, because it's the basic and the first level of immunity against invading cells and pathogens. On the other hand, we have the adaptive immune system. So this is performs a specific response to a particular pathogen or antigen. So technically, in this case, it's 1:1 response. So for every virus, for every bacteria, there's a specific response against them. Okay, so the adaptive immune system, okay, confers a lag time between the time of exposure as well as the response. So most of the time, it will need certain days, like 5 to 7 days, before you can actually see the adaptive response to a particular pathogen. So it also has cell-mediated numeral components for cell-mediated components. So that will be including B cells, okay, and T cells. Okay, for humoral components, most of the time they are the antibodies that are released by the mature B cells known as the plasma cells. So the exposure, okay, leads to immunological memory. So therefore, the some of the B cells and T cells confers a memory function, okay, that will be activated again if the pathogen as is that is previously been removed has been entered once again in the body. So this is found only in jawed vertebrates, okay, wherein certain tissues and organs like the bone marrow is already found kasi that is where the B cells, T cells um, originate. Okay, but for other types of forms of life, ayun, so the adaptive immune system is not observed. Okay, so here our focus is on adaptive immunity, okay, which bears the recognition on the specific non-self antigens. So when we say non-self, these are antigens or substances that are not usually found in your body. Okay, so this is in presence of the self antigens, which are technically substances that are coming from your body. Okay? So technically, the goal is to generate a pathogen-specific response, okay, that eliminates these specific pathogens or even the pathogen-infected cells, such as in the case of the viruses. Ayan. So also, it develops an immunologic memory that can quickly eliminate a pathogen upon a second exposure. Yeah. So that's why in most cases, for example, chickenpox, measles, and so if you have already been contracted with these diseases, ayun, when you are exposed once again to the pathogen causing these diseases, ayun, so even though you get the disease, ayun, it will last for a few days na lang or even hours because of the very fast response of the um, memory cells. Ayan. So this figure shows the different responses. Ayan. So we have here an antigen presenting cell such as macrophages, dendritic cells. Okay. So in the presence of an antigen, so they will process this antigen, okay, and present this to a helper T cell. So um, so the helper T cell will activate other cells, ayan, other T cells and B cells. Ayun. So in this case, since this is a CD4 helper T cell, ayun, it will activate other immune cells such as B cells then. Okay. Also, it can activate other cells like macrophages and killer T cells. Okay. So the difference lang of these cells is their function. So macrophages can engulf um, pathogens and virus infected cells, okay, and then neutralize them. Killer T cells, on the other hand, are T cells, okay, that when it binds to an infected cell, ayun, it will cause that particular infected cell to undergo apoptosis. Apoptosis is technically the program cell death. So technically, the normal programming of the cell, okay, in order um, that will eliminate self, ayun, will be turned on. Okay, when killer T cells bind to infected cells. Ayan. Tapos here at the top, we have the B cells, which as I mentioned a while ago, the B cells will be activated and form plasma cells. The plasma cells are the ones responsible for the formation of the antibodies. Okay. Ayan. So here, another illustration on the interaction between the cells. We have here an antigen presenting cell. Okay. We also have here the helper T cell. So the interaction is done using the class 2 major histocompatibility complex molecule and an antigen receptor here, or the T cell receptor. Ayan. So here's the antigen that may came from the pathogen itself. Ayan. So here, the helper T cell is activated, okay? And this activated helper T cell will activate other cells, such as B cell, releasing cytokines, okay, in the process. And then this B cell will become plasma cell that will release the antibodies. And some of a small fraction of these activated B cells will become memory B cells. Okay, so they will be the memory guard of our body. Okay, just in case another pathogen of the same kind, okay, will invade our body. So in that case, the response will be much faster. Okay, so B cells are involved in the creation of antibodies circulating in the body, in the blood plasma, as well as the lymph and the lymph vessels. Okay, so this is known as the humoral immunity, okay, or the adaptive humoral immunity. So antibodies are Y-shaped proteins, and these are used by the immune system to identify and neutralize certain objects, foreign objects, or objects and substances coming from the pathogen. So there are 5 isotypes of antibodies. We have the IgA, IgD, IgE, IgG, and IgM. So they have various properties. Okay, so for example, the IgG and the IgM are the first used, okay, by the body in combating diseases. Ayun. So we also have IgE, which has antiparasitic properties. IgA is antibody in the secretion, such as in the case of humus, in the case of mucosa linings, etc., etc. IgD is a type of antibody that is released also through secretions. Ayan. So upon activation, the B cells will produce these antibodies regarding considering the specific pathogen that the body is combating. So technically, these antibodies will neutralize a specific antigen. Okay, an antigen is a substance coming from the pathogen. Okay, so the antibody will neutralize the antigen. Okay, so glycoproteins belong to the immunoglobulin superfamily. They are also found in the blood, specifically bound to the B cells, which increases the activity of these antibodies. So this is the structure of the antibody. So this is the IgG to be specific. So we have here the Fab domain or antigen binding fragment domain. Ayan, we also have here the antigen binding site, which is similar to the active site of an enzyme. Ayun, tapos dalawa siya since Y-shape ito. So there's another antigen binding site here. And then we also have here the Fc domain or the constant domain. Okay? So ayun. So technically here in the Y-shape antibody, specifically in IgG, it can bind to at most 2 antigen, one here and one here. So also just to share information on the Fc region, so it is most of the time the amino acid sequences are similar to other antibodies. That's why it is known as constant region. Okay? Tapos we only have variation only at the antigen binding site. Okay, that will also confer the specificity. The amino acid sequences on this particular region confers the specificity of the antibody. Ayan. So when the antigen binds to the antigen binding site of the antibody, okay, it will form the antigen antibody complex that is shown right. So technically it looks like a substrate enzyme complex, parang ganun. Okay? So these particular complexes are produced upon the action of the humoral immunity. So here we have here the antigen antibody complexes. Ayan. So ayan, here's another illustration of the antigen antibody complexes. Ayun, so we have here different antigens on the surfaces of the pathogen. Tapos you have your antibodies that are bound on them. Ayun. So in most cases, these particular complexes may precipitate out, okay, from the mixture, okay, or this entails the easiness or easy removal of this particular pathogen. Okay. So how do we detect the presence of an antigen or even antibodies in vitro? Okay, so meaning in laboratory settings or using tubes, using different plates. Okay, so we can use enzyme labeled assays, okay, which are currently used and most widely used, okay, in order to investigate in immune responses and even activities. So because a single enzyme label can provide multiple copies of detectable species, and therefore can be used to identify reactivities. So enzyme immunoassays label either as ligands or antibodies with the enzyme, or an enzyme activity in bound or free fractions is measured using these assays. So ELISA, okay, the major activity, okay, for this experiment, okay, it uses antibodies, okay, that are exquisitely specific, okay, to a particular antigen. Okay, you can use this to detect or quantify the amount of protein or other antigen that is found in your sample. So this make use of an enzyme that react with a colorless substrate, and the complex, okay, will produce a certain colored product. So the enzyme here is covalently linked to a specific antibody, and this recognize a specific antigen. So if the antigen is present, the antigen antibody complex, it will bind to the enzyme, and on the addition of a substrate, the enzyme will catalyze a reaction generating a colored product. So later we will have an illustration for this. So the presence of a colored product indicates the presence of the antigen. So that, but though it is indirect, okay, but that means that when the color is observed, okay, so therefore the antigen is present. So it can detect less than a nanogram of a certain protein. So it's highly specific, okay, and very sensitive technically. So this may use polyclonal or monoclonal antibodies, okay, which are produced by multiple cells or a single cell, B cell to be specific. Okay, so here are examples of the indirect as well as the sandwich ELISA. Okay, so in letter A right here, so you have here your indirect ELISA. So here's your wells, okay, or glass wells that is coated with the antigen. So here's the antigen, the red one. So after washing with a certain buffer or any solvent, ayun, so what you will add next are your antibodies and allow them to bind to the antigen that is coated on the well. Okay, so technically, ayan, the antibody will bind to the antigen, and then upon washing again, another antibody, or in this case, we shall call it a secondary antibody, okay, will be um, directed towards the Fc region of the first antibody. Ayan, so the secondary antibody, okay, has the enzyme that is linked with it. Okay? Ayun. So when the binding is complete, ayun, you can now add your substrate S. Okay? Upon the reaction of the substrate with the enzyme, ayun, it will form a colored product. So the substrate is added, converted by the enzyme into a colored product. So the rate of the colored formation is proportional, of course, to the amount of the specific antibody present in the sample. Okay, so here you can use this to detect the presence of antibody, if there is. Ayan. In letter B, this is sandwich ELISA, which is similar to sandwich. So first is that your well is coated with a monoclonal antibody. So you start with an antibody here. So now you will add a sample or mixture containing the antigen. Okay, and if the antigen that is specific that is detected by the first or primary antibody is there, it will bind, creating the complex right there. Okay. Upon washing, an addition of the secondary antibody, Ayan, so this secondary antibody will also have a specificity to the antigen. Ayan, so creating the sandwich right there. The secondary antibody again is linked to an enzyme, and upon the addition of a substrate, it will cause the substrate to change its coloration or to have a color that can be observed visually. Okay, so ayan, so that's the difference between the indirect ELISA and the sandwich ELISA. Okay. So here we have so far we have seen the indirect and the sandwich. Okay, so just to have the other types of ELISA, we have here direct ELISA, wherein you have your enzyme that is linked to antibody, okay, directly interacting with the antigen present. So that's direct. Okay, so and also we have the competitive ELISA. Okay, so here we can add an antigen inhibitor. Okay, and upon addition of the substrate ion, it will compete, okay, with the activity with the reactivity of the antibody to the antigen. So here are some of these steps, major steps in the methodology. Okay, so these are also included in the manual. Ayun, so first is that you need to prepare your 96 well plate. Ayun, and then using sterile tips, you need to add your sample as well as the controls to the wells. Okay, so ayun. So these are all parts of the kit. Okay, and make sure to follow them properly. So ayun. So next, you will add the primary antibody. Okay, the antibodies will seek out the antigen from the many proteins that are bound to the well. Okay, so it will look like an indirect ELISA for us. So if your sample contains the antigen, the antibodies will bind to it and remain in the well. Okay, so the next is that you will wash it. Okay, so will wash it to remove excess antibodies. Okay, and then add the next secondary antibody, okay, with horseradish peroxidase or HRP labeled antibody. So this peroxidase is the enzyme E a while ago. Okay, and it is connected to the secondary antibody. So ayun, if the primary antibodies rather, have been bound to the antigen previously, so this antibody with HRP will bind to the primary antibody. Okay. So ayun. And then of course, you need to wash it to remove excess of the antibodies, secondary antibody. Okay. And then the next thing that you'll do is to of course add the substrate. Okay. So wait for 5 minutes in order to allow them to react, the substrate to react with the HRP, okay, or the peroxidase. Ayun, and evaluate the assay result. Okay. So in this case, if the antigen was present in your sample, the wells will turn blue. Okay. If this is a positive diagnosis or a positive result. So if the wells remain colorless, therefore the antigen is not present in the sample. Okay. Ayun. So applications lang of the ELISA is the detection of certain viruses such as the HIV. So HIV, or the human immunodeficiency virus, is a type of lentivirus, okay, a subgroup of the retrovirus actually. So these viruses contain retro enzymes, okay, that can return a particular mRNA to a DNA, etc., etc. So this is a sexually transmitted infection, okay, and this virus attacks the immune system, specifically the helper T cells. Ayun. So this reduces the CD4 plus helper T cells in the body, and when it progresses, it becomes AIDS, or acquired immune deficiency syndrome. Okay, so there's the photo of HIV together with the capsid, the envelope, the RNA, the glycoproteins, as well as the um, retroenzymes. Okay, so ayun. So here, in the detection of the HIV, so we use the HIV B24 antigen. Ayun. So this is usually observed, or highest peak, okay, 15 to 25 days upon the first exposure to the HIV. Okay. So therefore, at this particular time, okay, we can use different um, antibody. We can use the ELISA to detect the presence of the antibody. Ayun. So we have here at the fifth day, we can use the not detectable HIV RNA. Okay. And you can also use the P24 antigen as well. Okay. That is observable at daytime. But it peaks at the 30th day, in 1 month after the first exposure to the virus. Okay. So ayun. So this experiment um, applies the concept of ELISA. Okay, so you will need to check the biochemical and immunology principles in the antibody production, as well as discuss the principles of enzyme-linked immune sorbent assay or ELISA. Okay, and in the lab, you will perform the ELISA using the ELISA kit that we have, okay, for the HIV P24 antigen detection in the blood samples. And of course, appreciate the use of ELISA as one of the molecular techniques used in molecular biology and biotechnology. So ayun, so you will have your different tubes with you. Okay, so here you will have yellow tubes with each student's initials. So you may have put your initials in there. So label the outside wall of each well with of your 12 well strip. So two students may share a strip of 12 wells. Ayan. So on each particular strip, okay, label the first three wells as positive like this. And then the next three will be negative. Okay. So this is done in order for us to have at least three replicates for the positive and the negative. Okay. So ayun. So on the remaining wells, you may want to write your initials and your partner's initials. So for example, ayan, we have here Florence Nightingale and Alexander Fleming. For example lang naman. Okay. Ayun. So that is how you will label your wells. Okay. So the next is to bind the antigen to the well. So using a pipette or rather, transfer 50 microliters of the positive control to from the violet tube into the three positive wells. Okay. So please be cautious in transferring the controls as we have only limited volumes of the controls. Okay. So as much as possible, do not over push the pipetor, okay, in getting the control or dispensing them. Okay. So and then after each particular transfer, okay, so for example, you're done with the positive control, use a fresh tip to transfer the negative control naman. As much as possible, if you're going to change samples or changing controls, use a fresh or a new tip, pipette or tip, okay, in this case. Okay, and then of course, use another tip for the transfer of the samples and transfer each of 50 microliters of your team samples into approximately initial 3 wells. So, yeah, so wait for 5 minutes while all the proteins are sampled bind to the plastic wells. Okay, so give them time. Okay, so I think 5 to 7 minutes will do. Okay, and then after that, you will need to wash the unbound samples out of the wells. So to perform this, okay, so um, tip the microplates strip upside down. So turn it upside down, baliktarin. Okay, onto paper towels so that the samples will drain out. So literal na itatapon. So gently tap the strip a few times upside down. So literal na tataktakin na lang siya, but gently. Okay, gently do it. Okay, and make sure to avoid the splashing of samples back into the gas, as in mag-transfer, let's say from positive, it goes to the negative. As much as possible, prevent or avoid that from happening. Okay, and then after that, you will need to discard the top paper towel. Okay, so next is for you to fill the wash with a pipette with a wash buffer. Okay, and fill each well. So this will be the washing part. Okay, so after you have added enough buffer to each particular well, so tip the microplate strip upside down ulit and wash it and drain it out. So discard the top 2 to 3 towels because they may have contained the buffer. Okay, so you're done with the first washing. Then the next thing that you're going to do is, of course, repeat it. Repeat the washing once again. Repeat the washing once again, at least twice. So to make sure that all of the excess antibodies has been removed. Ayun. Then use, all of the excess samples has been removed. So the next thing you're going to do is to pipette the primary antibody or the PA from the green tube into the 12 wells. So primary antibody to each of these. So again, make sure that you will have 50 microliters lang of the primary antibody as much as possible. Okay? And then after the addition to the last well, wait for 5 minutes for the primary antibody to bind. Okay, so just leave it as is. Okay, and wait for 5 minutes. Okay, and then repeat the washing once again. Okay, so dip the wells into a paper towel. Okay, and then remove the solution, and then wash it again with the buffer. Okay, repeating the washing process a while. So the next step after the twice washing is for you to add the secondary antibody. Okay, so this secondary antibody is coming from the orange tube, okay, and transfer it to the 12 wells. Okay, so once again, okay, after addition, we wait for 5 minutes. Okay, allow. So wash the unbound antibody. Okay, out of the wells, and then repeat the washing the washing step 3 times. As much as possible, repeat it 3 times, okay, in order to ensure that everything is removed. Okay, so next is for you to add the substrate, okay, from the brown tube, okay, and add them to the 12 wells, okay, and then again, just like what you're doing last time, so wait for 5 minutes and observe for the results. Okay, so here's a summary of the tubes, okay, their contents, okay, and their particular number, okay. Ayan. So precautions lang on the this particular experiment. Again, proper pipette tip use and proper micropipettory use. Okay, because we have a limited samples, we have limited amount of the enzymes, substrates, etc. Um, please be careful in handling in getting as well as transferring them. Okay? Okay. And then proper microtiter plate washing, of course, addition of the buffer and then removing it. Okay, so make sure that you have done that. And then of course the reagent use as much as possible. Use the proper reagents, okay, from each specific colored tubes only. And then of course observe incubation periods correct. Okay, so these are the references, and this ends um, the discussion. So thank you, and good luck in the experiment. Thank you.